All right, welcome to our super exciting lesson on mitosis, which is a fancy word for cell division. You will probably need to pause this video along the way. I am going to go quickly. It's all important, and I will highlight the super important stuff. We watched these in class. If you didn't, go back and watch them. So the whole point of mitosis is why do multicellular organisms like you divide? And it is for these three reasons. You need to get bigger, you need to fix your owies, and you don't want to look like a 2-year-old, so we have obviously had cell division to develop your body into a human. The fourth reason that our cells divide would be to make sex cells, and that is a process called meiosis, which we will talk about next. So we're going to talk about cell division for these three reasons up here. So basically, you started out as an egg and a sperm. That original fertilized egg is called a zygote. That zygote divided millions of times, and those cells specialize. It's a pretty cool process how that happens, but they do specialize, and they made a multicellular organism, which is you. If this didn't happen, you would be a single-celled organism. You would be an amoeba, a paramecium, euglena. These are eukaryotic cells. They have membrane-bound organelles and a nucleus, but they're a single-celled organism. Okay, quick review of those two cell types, prokaryotes versus eukaryotes. So again, we're going to focus on the eukaryotes, but a quick look at prokaryotic cell division. Remember, those are single-celled organisms. They don't have a nucleus or membrane-bound organelles. It is limited to our bacteria groups, and their cell division is called binary fission. That's actually how they reproduce. So they do cell division similar to mitosis, but it's not mitosis because they're not doing it to grow or repair or develop. They're doing it to reproduce. So we call it binary fission or asexual reproduction. So again, it's similar to mitosis, but because it's for reproduction, it gets this different name. So prokaryotes grow by getting bigger, eukaryotes grow by making more cells. Why don't prokaryotes get as big as eukaryotes? Can you imagine a single-celled organism as big as this guy? I can't remember his name. Just one big amoeba. So there are reasons. So surface structure and what we talked about in that cell size lab, getting nutrients in and waste out. So this is a review of that cell size lab. Hopefully you know that. Now we're going to talk about our eukaryotic cells who divide for growth, development, and repair. So remember, eukaryotes are multicellular organisms. They do have a nucleus, they do have membrane-bound organelles, and it includes everything that is not bacteria. So please make sure you know the difference. Eukaryotic organisms multiply by sexual reproduction, and that's the process of meiosis. Prokaryotic organisms multiply by binary fission. So lots of words here simply to remember. You will reproduce someday with sexual reproduction. Bacteria reproduce, and they call it binary fission, and it's similar to mitosis. All right, let's go. So here's another look. Meiosis creates the sex cells. The sex cells get together. They make a zygote. The zygote divides and divides. It's called an embryo. It's called a fetus. It's eventually called a baby, and that is how that happens, and then it turns into a human, and that is a really cute baby. So here is another look at mitosis before we start. Mitosis happens in somatic cells. Remember, somatic, the word soma means body, and that is opposed to a sex cell. So we are talking about your body or your soma cells, which means they can be somewhere or anywhere in your body. That's how I remember that. Somatic cells are everywhere or somewhere, and when they divide, DNA has to replicate. That's what we talked about before Christmas, so bring back all those memories, or maybe right after Christmas. DNA makes a copy of itself and it turns into this X. So this side of DNA split open, it copied, it's that semi-conservative anti-parallel held together right here at a centromere. So DNA divides, they line up in the middle, and that's not a very good picture of lining them up, but then we rip them apart at that X, and one half or one side of that replicated DNA goes into each of the two new cells. So I'm going to talk about this process in detail right now. Quick review of what DNA is. This is super important to me that you know this. This down here is a strand of DNA, okay? It's all the nucleic acids, but it can't exist alone. So DNA is actually called a double helix because those nitrogen bases bond together. They cannot exist by themselves. Those hydrogen bonds are too reactive, and they just bond onto that other side. So DNA double helix is what we call DNA. I want you to know that it's made of 2 single strands. That's why it's called double. When we coil that up, the only way you're going to hold that coil together is by throwing some proteins in there called histones and nucleosomes. So when DNA is coiled up, we give it a different name because we've added these proteins. So coiled up DNA is called chromatin. The only time we have chromatin coiled like this is after replication. So every time you see a chromosome, they're usually shown looking like an X. But again, this X is replicated DNA. Here's a review of that stuff before Christmas. You had your original strand of DNA, helicase split it open, DNA polymerase added the new side. You end up with 2 identical strands that are semi-conservative but completely identical, and they are actually held together like this. So when you see a chromosome that's an X, it is not a crisscross X. It's this line and somebody pinched them together in the middle and put a belt around them. So there's a strand here and a strand here. It's not crisscrossed, which I think is important to understand. So this is replicated DNA. This is a chromosome, but now this chromosome has a second double helix, that identical copy. Super important to know this. And again, we're going to call this a chromatid and this a chromatid. They're made of chromatin that's coiled up to be a tid, and the whole thing is a chromosome. I will not test you on that, but I do think if you plan on going on in bio, you should learn all that vocab. Okay, a little bit more terminology. So chromatin is before DNA gets coiled up. Chromosomes are when they are replicated and they are that X. We have homologous pairs. Remember, the prefix homo means the same. So this would be chromosome 1 in your karyotype. So if you remember those karyotypes, it had 2 chromosomes. If you look at those karyotypes closely, each chromosome is made of replicated DNA. This chromosome number 1, these are homologous chromosomes. So this person, there's a gene here they got from mom and a gene over here they got from dad. That's why they're homologous chromosomes. But they're actually made of replicated DNA, so we call that a tetrad because there are 4 strands of DNA here. So here's what it looks like. Whoops. Oh, I thought I had some pictures here. This is that picture of your chromosomes. This is a homologous pair. So this is what I showed you on the other slide. These look like a single strand, but this is actually replicated DNA. There's an X right here and there's an X right here. So when you see this picture, yes, there are 46 chromosomes. Each chromosome just happens to be made of replicated DNA. All right, that was a lot of vocab. Let's go on to what's actually important, which is the cell cycle. The cell cycle is interphase, mitosis, and cytokinesis. So remember, these are different phases. Interphase is not mitosis. It is when the cell is doing its thing. So if you think about your life, you were born and you grow up and you do your job and blah, blah, blah. And then you have a really short time in your life when you're going to reproduce. And then your baby grows up and does its job. So most of your life and most of a cell's life is just doing its thing. It's doing protein synthesis. At the very, very end, when that cell gets to a certain size, it will divide in half through mitosis. So in interphase, what you need to know, this is when DNA replicates itself. It starts to coil up. Not fully, but DNA replicates and starts to coil up to form that chromosome. Then we get to mitosis. So mitosis has 4 subphases: prophase, metaphase, anaphase, telophase. PMAT is how I remember that. So remember, this is different. Interphase is not part of mitosis. So the DNA was replicated in interphase. Now we get ready for mitosis. And again, the way to remember that is PMAT. Lots of things happen in prophase. It's actually the longest phase of mitosis because the chromosomes finished condensing. We get these little centrioles that appear out in the cytoplasm, and they have legs coming off of them called spindle fibers, and the nuclear membrane disappears. So everything's getting ready here. We've got the chromosomes replicated and they're condensing so they can fit. The centromeres and spindle fibers are kind of like our fishermen and the fishing line. They're going to move to either side of this cell and eventually these spindle fibers hold on to the chromosome and pull it apart. So it's like a fisherman right here, and this is his fishing line, and they're going to do the work. So lots of stuff happens here. This is what it looks like in real life. The chromosomes look like little worms, but you can kind of see the outline of that cell membrane, and we cannot see the centrioles here because they're just too tiny, but they're there. Metaphase, so nuclear membrane is totally gone. The centrioles are down at either end, and they have sent their spindle fibers and caught their fish in the middle. So notice right here, those homologous pairs are lined up head to toe. So this could be the chromosome from mom. This is one from dad. Here's chromosome 2 from mom. Here's chromosome 2 from dad. So they line up head to toe across the middle. This is an important thing to remember. The spindle fibers hold on to one side of that replicated DNA. So metaphase, we're getting in the middle. That's it. Line up in the middle. M for middle, M for meta. Anaphase is kind of a violent phase. So if you have a friend named Anna, pretend that she is violent, because now we're going to rip apart that replicated DNA. So remember, these two strands were part of that X. They were made with DNA replication. They're identical to each other, held together at the centromere, and the spindle fibers are ripping apart that replicated DNA. So this organism that we're talking about here had 1, 2, 3, 4 chromosomes, so 2 from mom and 2 from dad. When you rip them apart, 1, 2, 3, 4 double helixes go this way, and 4 go that way. These are copies of each other. So you replicated the DNA. This little line here is exactly like this little line. So they're going to opposite ends of the pole, being pulled by the centriole and the spindle fibers. The end is telophase. So it's the opposite of prophase. If you can remember what happens in prophase, just undo it. Nuclear membrane gets reformed, chromosomes relax, and spindle fibers and centrioles are going to disappear. This is the beginning of a cell plate getting ready to divide this animal cell, and that's what this is up here. So at the beginning of telophase, you're going to see chromosomes condensing, nuclear membrane reforming. If it's an animal cell, the cell membrane pinches in, and this is called a cleavage furrow. So cleavage, just like how we talk about female parts, it looks the same. It's a cleavage furrow. Okay, the word just means a pinching in. That's why we use that word cleavage. Late in telophase, all this stuff is formed, and kind of at the same time, we have the dividing of the cytoplasm, which is cytokinesis. But cytokinesis is its own thing. It's not really part of telophase. At the very end, you need to know this. 2 daughter cells, so we use this female pronoun here, I don't know why, but you do, come from that mother cell. Again, it's a female or parent cell, and it has the full set of DNA. So you have made an exact copy. We replicated the DNA, we lined it up, and we split it apart. Here's another picture of telophase in a plant. There's a cell wall. Here's an animal, that cleavage furrow. All right, here's where it gets kind of fancy. I want you to think about this. Here's an original cell, okay? There are 46 double helixes of DNA right there. After replication, so after interphase, that DNA is doubled. I had to pause there. I don't know what I was saying. After interphase, DNA is doubled. You can see these X's. So this organism has 1 chromosome. It got the red one from mom, the blue one from dad. It replicates that DNA. They line up head to toe. Anaphase rips them apart where they were replicated, and you have 2 daughter cells that look exactly like what you started with. So for a brief moment there, you have 92 strands of chromosomes, but they're glued together, or 92 strands of DNA as 46 chromosomes. At the end, or right when they're splitting, there are 92 chromosomes. They're called a chromosome now because they're by themselves, but then at the end, you end up with that original 46 strands. Okay, here are some pictures. I think you should pause here and get used to looking at that. So chromosomes condensing, here's metaphase, here's anaphase and telophase. This is a picture in a plant cell, so pause here, make sure you can identify those. And that is as far as we are going to go today.